The immune system is the body's natural defense mechanism that protects us from harmful microorganisms such as bacteria, viruses, fungi, and parasites. It works continuously to identify and eliminate anything that is considered foreign or dangerous to the body. This system is made up of various cells, tissues, and organs that work together in a coordinated way. Some of the main components include white blood cells, also known as leukocytes, which play a major role in fighting infections. In addition, organs such as the bone marrow, thymus, lymph nodes, and spleen are also involved in producing, storing, and activating immune cells. Overall, the immune system is essential in maintaining our health and preventing from. The immune system functions through three important stages, which are reconnaissance, recognition, and response. First, during the reconnaissance stage, immune cells constantly patrol the body, monitoring tissues and the bloodstream for any signs of infection or foreign invaders. This allows the immune system to detect potential threats early. Next is the recognition stage. In this stage, immune cells identify specific molecules on the surface of pathogens called antigens. This step is crucial because it helps the immune system distinguish between the body's own cells and harmful foreign substances. Finally, the response stage occurs when the immune system activates different defense mechanisms to eliminate the pathogen. This may include inflammation, phagocytosis, where immune cells engulf pathogens, or the production of antibodies to neutralize the immune system can be divided into two main types, which are innate immunity and acquired immunity. Innate immunity is the first line of defense and is present from birth. It responds quickly to infections, but is non-specific, meaning it reacts in the same way to all pathogens. Examples of innate immunity include physical barriers such as the skin, as well as immune cells like macrophages that destroy pathogens through phagocytosis. On the other hand, acquired immunity develops after exposure to a specific pathogen. This type of immunity is highly specific and involves immune cells such as B cells and T cells. Although the initial response is slower, acquired immunity provides long-term protection through immune
Next, I will be explaining about innate immunity, focusing on the first line of defense. Innate immunity is a non-specific defense system that is present from birth and provide immediate protection against pathogens. The first component of the first line of defense is the skin. The skin acts as physical barrier that prevent bacteria and pathogen from entering the body. Under normal condition, microorganisms cannot infect skin. However, small cuts or abrasion may allow pathogen to enter the body and cause infection. The second component is mucous membrane. Mucous membrane line the respiratory, digestive, and reproductive tract. This membrane secrete mucus which trap microbes and particles. For example, mucus in respiratory tract help prevent microorganisms from reaching the lungs. The third component is body secretions. Tears and saliva contains an enzyme called lysozyme, which can destroy the cell walls of certain bacteria. Sweat and oil secretion makes the skin surface acidic, which inhibits microbial growth. In addition, stomach acid kills many microbes that enter the body through contaminated food or water.
Today, I will explain about innate immunity, focusing on the second line of defense. So, how does the second line of defense work? The second line of defense provides non-specific protection against invading microorganisms. It acts after pathogens that pass physical barriers and involves several internal defense mechanisms. White blood cells also plays an important part by ingesting and destroying microorganisms. These lines of defense also include inflammations, antimicrobial proteins, and specialized immune cells that work together to prevent the spread of infections. The second lines of defense also include cellular innate defenses, which consist of specialized immune cells. These include phagocytic cells and natural killer cells, also known as NK cells. Each of these cells has a specific role in protecting the body from infections and abnormal cells. For phagocytic cells, they function by engulfing and destroying pathogens and cellular debris. They attach to pathogens using surface receptors, form a vacuole around the pathogens, and then fuse this vacuole with lysosome for digestion. Natural killer, on the other hand, patrol the body and target virus-infected cells and cancer cells. They do not attack microorganisms directly, but instead they release chemicals that cause infected or abnormal cells to die, helping to prevent further types of phagocytic cells. First is neutrophils. Neutrophils make up about 60 to 70% of white blood cells. They are attracted to infected tissues where they engulf and destroy invading pathogens, and they often self-destruct after completing their function. Next is monocytes. Monocytes make up 5% of white blood cells. They circulate in blood for a few hours before migrating into tissues, where they differentiate into macrophages. The third one is macrophages. Macrophages are large and long-lived phagocytic cells. They migrate throughout the body and are found in various organs of the lymphatic system, where they play a key role in destroying pathogens. The fourth one is eosinophils. Eosinophils make up about 1.5% of white blood cells. They are commonly found beneath mucosal surfaces and attack pathogens by exocytosis of toxic compounds and by releasing a destructive enzyme. Last but not least is dendritic cells. Dendritic cells are located in tissues exposed to the environment, such as skin. They act as phagocytic cells, and their primary role is to stimulate the development of acquired immunity, linking innate and adaptive immune responses. Next is the process of phagocytosis. The process of phagocytosis begins when pseudopodia surrounds the microbes. The microbe is then engulfed into the cell, forming a vacuole. This vacuole fuses with lysosome and enzyme along with toxic compounds to destroy the microbe. Finally, the debris is released from the cell by exocytosis. Next is the lymphatic system. The lymphatic system plays an important role in innate immunity. It collects interstitial fluid to form lymph, which flows through the lymphatic vessels. Lymph nodes filter pathogens, and immune cells such as macrophages, dendritic cells, and lymphocytes respond to invaders. Finally, the lymphatic system returns lymph to the bloodstreams, maintaining fluid balance and immune protection.
Moving on, we have antimicrobial protein, which act as chemical reaction defense. And antimicrobial protein is divided into two types, which is complement system and interference. For complement system, it helps the activation of phagocytes such as macrophages or neutrophil, making it easier for them to recognize and engulf those pathogens. This process increases the efficiency of phagocytosis. And secondly, it also complements system trigger inflammation. Inflammation helps to increase blood flow and capillary permeability at the infected area, allowing immune cells to reach the site of the infection more quickly. And lastly, complement system also can cause pore formation on the surface of the pathogen, which can lead to lysis and burst. As for interferon, it's a protein released by the body cell when they get infected by those viruses, which their main function is to protect nearby cell from becoming infected. So, they will inhibiting the viral replication, which means they prevent viruses from multiplying inside those host cells. In addition, interferon also activates macrophages and natural killer cell, which is activation of macrophages will help in engulf and digest those viruses, while the NK cell, which is called also known as natural killer cell, will destroy the virus infected cell directly. This helps to limit the spread of viral infection within the body. Overall, interferon also play an important role in innate immunity by slowing down the virus spread and enhancing the activity of immunity. And as for final and last component of second line defense in innate immunity, we have inflammatory response, which is a response occur when the body tissue are injured or having infected by pathogens. The cell that damaged will release chemical substance that called histamine. Histamine plays important role in inflammation as it can cause vasodilation, which means the blood vessel itself will be widened and allowing more blood to flow to the injured area. In addition, histamine also increases the capillary permeability, which allow white blood cell, antibody, and the plasma protein to leave the bloodstream and enter the affected tissue more easily. As a result, immune cell can quickly reach the site of the infection or the injury. The increased blood flowing and fluid leakage also help to isolate the eliminated pathogens as well as begin tissue repair. However, these changes also lead to classic signs of inflammation, which is heat, pain, redness, and even swelling. In conclusion, the inflammatory response is an essential part of innate immunity.
Acquired immunity. Acquired or adaptive immunity is a type of immune response that develops after exposure to specific antigens. Unlike innate immunity, this response is highly specific and involves specialized immune cells called lymphocytes. The two main lymphocytes involved are B cells and T cells. A key feature of adaptive immunity is its ability to remember previous infections, allowing a faster and stronger response in pre-exposure. B cells are lymphocytes that mature in the bone marrow and play a major role in humoral immunity. When a B cell recognizes its specific antigen, it becomes activated and differentiates into plasma cells and memory B cells. Plasma cells produce antibodies that bind specifically to antigens, helping to neutralize or eliminate them. Memory B cells remain in the body and provide long-term immunity. T cells mature in the thymus and are responsible for cell-mediated immunity. Unlike B cells, T cells do not produce antibodies. Helper T cells, also known as CD4 positive T cells, assist in activating B cells and other immune cells. Cytotoxic T cells or CD8 positive T cells directly destroy infected or abnormal cells. T cells recognize antigens only when they are presented on major histocompatibility complex or MHC molecules. Antigens and epitopes. Antigens are foreign substances that trigger an immune response in the body. This may include bacteria, virus, or proteins. Each antigen contains specific regions known as epitopes, also called antigenic determinants. Epitopes are the exact parts of the antigen that are recognized by immune receptors on B cells and T cells. Different immune cells can recognize different epitopes on the same antigen. Clonal selection and immunological memory. Clonal selection occurs when a specific B or T cell binds to its matching antigen. This causes the cell to rapidly divide and produce many identical clones. Some of these cells become effector cells that fight the infection, while others become memory cells. Immunological memory allows the immune system to respond more quickly and effectively if the same antigen is encountered again. Humoral versus cell-mediated immunity. Humoral immunity is mediated by B cells and antibodies and mainly target extracellular pathogens such as bacteria and toxins. In contrast, cell-mediated immunity is mediated by T cells and targets intracellular pathogens such as viruses that infect host cells. Both types of immunity work together to provide complete protection against infections. Active immunization. Active immunization occurs when the body is exposed to an antigen and produces its own immune response. This can happen through natural infection or vaccination. Vaccines stimulate the production of antibodies and memory cells without causing disease. As a result, active immunization provides long-lasting protection. Passive immunization. Passive immunization involves the transfer of preformed antibodies from another source. This type of immunity provides immediate protection but is temporary because memory cells are not produced. Examples include maternal antibodies passed to a baby through the placenta or breast milk, and antibody injections such as antivenom.